Hello, I'm Adebisi Ogunsola. In this presentation, we also have Roha Chauhan and Folashade Adeboyejo. The research paper that we're presenting is based on the differential expression and signaling activation of insulin receptor isoforms A and B. The aim of the research is to find out if there is a potential link between breast cancer and diabetes. The study was conducted in paired samples which involved 100 normal and tumor patients from African American and Caucasian women. The range was between 20 to 90 years of age, which included 23 normal African American and 20 Caucasian. There were 27 African American and 24 Caucasian malignant. Death rates of African American women is higher than Caucasian women when looking at the insulin receptor expression and IGF-2 activation of the receptor signaling pathway in cell lines and breast tissues. Several factors are being examined as causative agents, such as variants in healthcare access, biological factors, and dissimilar use of screening tools. Higher IGF-2 stimulates tumor growth by signaling through IGF-1 and insulin receptor. As IGF-2 activates insulin receptor, the research proposes that African American has higher insulin receptor activation.
Hello, my name is Roha Chauhan and I will be talking about the techniques used in this research. There were overall 4 techniques used, which included real-time PCR, Western blot, immunohistochemistry, and phosphoELISA. PCR, which stands for polymerase chain reaction, is a biochemical technology used in molecular biology to amplify a few drops of a DNA or RNA to thousands and millions of copies in only a couple of hours. PCR does this by mixing the DNA with primers on either side. Fairly recently, a new method of PCR quantification has been invented. This method is called real-time PCR. It allows a scientist to actually view the increase in the amount of the DNA as it is amplified. There are several different types of real-time PCR being marketed to the scientific community at this time, each with their advantages. In this research, PCR application was performed using an iCycler bioread. Western blot, or sometimes called protein immunoblot, is an adaptation of the southern blot. Identification of the Western blot is based on molecular weight and antibody binding specificity. As the proteins migrate through the gel, they are separated based upon their size and charge. Characteristically, smaller proteins migrate through the gel faster than the larger proteins. There were 2 primary antibodies used in the Western blot. First was an anti-insulin receptor alpha subunit rabbit polyclonal antibody, or 5D9, and the second was purified rabbit polyclonal anti-human insulin receptor B antibody, or Y243. Biotinylated was used as a secondary antibody. Insulin receptor immunohistochemical analysis was performed using 2 different antibodies. One was 5D9, which was also used in the Western blot, as recognizes the alpha subunit of insulin receptor A and insulin receptor B, and purified rabbit polyclonal anti-human insulin receptor B antibody, as this recognizes the alpha subunit C terminal of the insulin receptor B, or Y243. The last technique, phosphoELISA technique, during this phosphorylation, the cells were treated with insulin growth factor 2 and or insulin. The PBDF membrane were incubated with antibodies against insulin substrate 1, phosphoinsulin substrate 1, and FAK and PFAK. Phosphoinsulin receptor was used and the reaction was visualized by adding T and B substrate. Now my colleague will be discussing the results that were achieved in the experiment.
My name is Folashade Adeboyejo, and in this graph, they analyzed the results of insulin receptor protein, especially in breast cancer cell lines of African American and Caucasian women. Estrogen receptor positive cell lines, MCL7 and CRL23T9 of African American, and the estrogen receptor negative cell line, CRL2335, express higher level of insulin receptor protein compared to Caucasian insulin receptor negative HS578T cell lines as shown in figure 1A and 1B. Estrogen receptor negative cell lines of Caucasian women had higher level of insulin receptor B when compared to African American breast cancer cell lines. They later analyzed basal levels of insulin receptor polarizations of trison 1160/116, which stimulates receptor kinase activities. The figure 1A shows higher polarization of insulin receptor for AA, African American insulin receptor positive cell line 2329, so as compared with Caucasian insulin receptor positive MCF7 cell lines. No basal level was deducted in Caucasian insulin receptor negative cell line HS578T. The bar graphs in figure 1B represent 3 or more experiments for each cell line studied. The beta-actin was used as a loading control. Now, I will be looking at the figure 3, which shows the comparison of insulin receptor expression and phosphorylation in cell lines in paired breast tissues of samples as assessed by using real-time PCR. By looking at the figure 3A and B, it can be clearly seen that the African American have higher level of insulin receptor A for normal and milligram tissue samples. In contrast, Caucasian women have more insulin receptor messenger RNA than African American for paired samples in pre and post menopause.
To conclude, the result clearly shows that high insulin receptor A and low insulin receptor B contributes to higher proliferation and lower metabolic response in African American breast cancer patients, as seen in figure 1. So different expressions of insulin receptor isoforms represent a potential biological link between breast cancer and diabetes. Insulin receptor substrates and focal adhesion kinase 1 activation also significantly increased in African American and Caucasian women.
Thank you for listening. That's it.